Hello everyone, can you all hear me? Am I audible? Is everyone here? Can we start? Everyone, please turn off your mic. So today our lecture is going to be, the first lecture is going to be from microbiology. So in the afternoon session, you'll be having PSM. So time is 8:30 now. So I'll try to comprise it within 3 hours. Okay, so before jumping into microbiology, there's something, yesterday we saw pathology. So there is something that connects pathology and microbiology, that is the immune system. So this topic, this notes is made in a way that any question that is asked from the immune system, regardless of pathology or even microbiology, you should be able to get it. So this notes will help in that. So I thought it's better we'll start from immune system before jumping into the microbiology. So this immune system is a combination of both patho and micro. So we are only quickly going to see an overview because I can't waste a lot of time in this. Okay, just listen quickly. So we'll go through this very fast. So basically, immune system is the one which protects us from not just diseases, from anything. So immune system is of two types. Yesterday we saw innate and adaptive. Innate is immediate, that occurs within minutes. So it's very speed in onset, but there is no memory. Whereas adaptive is you get from some problem, so you get adapted from it, so there is memory, so for the future. So all you need to know is memory is present only in adaptive. So adaptive means there is memory. So that's the first point that you need to know. Then, so yesterday we said innate is of a lot of types. It could be simple barriers like stomach's pH, GI mucosa, epithelium, normal flora. It could be inflammation, the local inflammation process that is happening, that is innate immunity. Phagocytosis by WBC, that is innate immunity. Our body's complement system, that is innate immunity. The cells like natural killer macrophages and monocytes, that is innate immunity. Cytokines, some cytokines, mostly some cytokines are innate, but very few are adaptive. So let's take out the cytokines from adaptive. So adaptive means you need to remember T cells and B cells. So that's the second point I'm going to say. So adaptive means memory. That's the first point. Adaptive means T or B cells. That is the second point. So anything other than this T and B cells, it is going to be innate immunity. For your MCQ, you need to strictly follow these two. I repeat these two MCQ. Adaptive means there is memory. Adaptive means T cells and B cells. So please make a note of this point. So I hope you may note at this point. Now, so whenever you get a, whenever your body is exposed to a pathogen, so our body finds two ways to destroy that pathogen. So the first way is called humoral immunity, which means attacking the organism indirectly. The second is cell-mediated immunity, which means directly you will attack the, or your body will attack the pathogen. So directly means it is going to swallow it, that is phagocytosis, or cytotoxicity is going to toxicate the cell and it's going to kill it. So you either eat it or you kill it. That is a direct mechanism. So cell-mediated immunity means phagocytosis and cytotoxicity. So humoral means indirectly, which means they are going to produce antibodies. So you need to remember humoral means antibody. So to remember this, I want you to write like this, H. So make a Y over here. This is the symbol of the antibody. So the antibody, humoral means antibody. That's the strongest point I want to say regarding your MCQ. Humoral immunity is antibody. Someone's microphone is on. Please turn it off. Everyone, please turn off your microphone. I don't want any interruptions here because it's a recording session. Okay, so humoral means antibody. That's the point. So humoral means, again, I want to say, you can make this as I. That is indirect killing. So humoral means indirect killing. I for indirect killing, Y for antibody. Indirect killing and antibody. So if you remember this, you can crack this MCQ. So obviously cell mediated becomes phagocytosis and cytotoxicity. So cell mediated means cytotoxicity. Cell mediated means cytotoxicity. And cell mediated means eating, that is phagocytosis. Eating, that is phagocytosis. Okay, then now, so there's going to be a target for both of this. So when it comes to humoral immunity, indirectly attacking, it is going to target mostly bacteria, that is extracellular pathogen. But when it's cell mediated immunity, it is going to, whatever is happening is going to happen within the body. So intracellular pathogens it will focus on, that is the viruses. So humoral means bacteria, cell mediated means virus. That is the next point. Humoral means bacteria, humoral means bacteria, humoral means bacteria, HB. So remember this HB pencil that we used to use in the childhood. So HB pencil, humoral means bacteria. That point you need to remember. Then cell mediated means virus. So remember, whenever you're going to an interview, you're going to submit your CV. So remember that CV. So cell mediated means virus. So you are going to use HB pencil to fill your CV. Just CV form. Just remember this point. HB pencil you will use to fill your CV form. This is the mnemonic I'm giving here. So humoral means bacteria. Humoral means bacteria. Cell mediated means viruses. They attack the viruses. So that's the primary target. Then humoral, the HB is not only for bacteria. Another B unit, remember, that is B cell, because we said antibodies. The precursor of antibody is going to be plasma cells, the precursor of which is going to be B cells. So again, this B unit, remember. So if you remember HB, your exam should go smooth. So HB, B is for B cell produces plasma cell produces antibodies. Okay, whereas CMI is for T cells, cell mediated cytotoxic, so T cells. CMI T cells, humoral B cells. HB means C is going to be T. Okay, I hope we are clear here. So in T helper cells, there are two types of T helper cells. Yesterday we talked about these two types, TH1 and TH2, but we were not able to go into detail because I thought I can cover this immune system again separately. So here's what we got. So T helper cell type 1, T helper cell type 2. So type 1 is going to be cell mediated, type 2 is going to be humoral. That is our antibody mediated. So this point you need to know. So most of the time cell mediated is going to be TH1. So most of the time cell mediated is going to be T cells. Okay. Why this TH2 is humoral and antibody mediated? Because its primary function is activating the B cells. That's why. So it activates the B cell. So you need to know the first type of T helper, that is the first receptor TH1, when it's activated, so it secretes cytokines which are interleukin 2 and interferon gamma, whereas TH2 is for interleukin 4 and 5. So you need to remember 1 is for 2, interleukin 2, 2 is in TH2 is for 4 and 5. 4 and 5. 4 and 5. To remember this point, you can simply think that you can just double it. So 1 means you can double it, 2 means you can double it, 4. So 5 will be a bonus. So 1 is 2, 2 means 4. So TH1 is interleukin 2, TH2 is interleukin 4. So 4 means you have to add 5 along with it. Okay. Okay. Next is immune organs. So immune organs means there is primary organs, there is secondary organs. So primary organs means where they mature. where the T cells and B cells mature. So T cells mature in thymus, B cells mature in bone marrow. So T for T, B for B. Okay, so only one point you need to remember other than that is B cell also matures in the fetal liver. So liver of the fetus, so fetal liver. So T cell thymus, B cell bone marrow, T for T, B for B. Other than that, B for L, fetal liver. Okay, next. In that point also you can remember HB, because hepatocytes, B cells, hepatocytes, B cells. So B cells, hepatocytes. Okay, B for BB, B for HB. Next is secondary. So the secondary immune organs can be lymph node, spleen, and MALT, mucous association lymphoid tissue. So lymph node is where the immune response is initiated. Spleen is the site of immune response to antigens in blood. Mucous associated lymphoid tissue could be from intestines or tonsils. So I'm not going much inside this. Now we have major cells in immune system. They can be neutrophils, eosinophils, basophils, mast cells, macrophages, dendritic cells, B cells, plasma cells, helper T cells, cytotoxic, memory, natural killer cells. So this many number of cells you have. So you need to know what they do. Neutrophils, they are going to eat the bacteria, phagocytosis. Eosinophils, allergy and parasites. So neutrophils are going to go for phagocytosis. So they're just going to eat the microbe. Eosinophil means you need to remember two things. One is allergy, another is parasitic infection. Allergy, parasite. So we already saw these points. Again, mast cells, there is basophil. Mast cells release histamine. This also we saw yesterday. Mast cells, basophils release histamine. Mast cells release histamine. Then comes macrophages. So macrophages work similarly like neutrophils. So they are going to eat up the microbes. So phagocytosis again. So forget about this cytokine point. Okay. Then comes dendritic cells. These dendritic cells present antigen. Only one point I want you to remember. Dendritic cell is called antigen presenting. Okay, its function is antigen presenting. Dendritic cell, antigen presenting. So where this is going to present? So this is a lot of MCQs actually. So a lot of times asked MCQ. Yeah. So you must know. So you must know dendritic cell present the antigen to T cells. B cell present the antigen to MHC type 2 restricted T cell. MHC type 2 restricted T cell. Okay, so this point is very, very important. So these two are presenting. So dendritic also presents, B cell also presents. So dendritic presents antigen to T cells. B cells presents antigen to MHC2 restricted T cells. Okay, MHC2 restricted T cells. So in any way they are going to present to T cells only. In any way they are going to present to T cells only. That's the point you need to know. So whatever it is, any antigen is going to be presented to T cell. That is the MCQ. Okay. Okay. So the others are not much important. So you need to know helper T cells means MHC, recognize antigen associated with class 2 MHC. Cytotoxic T cells means class 1 MHC. So class 1 MHC. But natural killer, they don't care about the MHC. MHC independent killing. So MHC independent killing. So in between there is something called memory cells. It could be for the B cell, it could be for the T cells. So the memory is for memory, which means for further in future, if there is any exposure to the same antigen, it is going to kill quickly. So the response would be quick. So more rapid secondary response. Okay. So that is easily understandable. So you need to know helper T, class 2, cytotoxic T, class 1, natural killer. They don't care about anything. They just kill. the tumor cells or virus infected cells. So that's it about this table. So very important. I repeat this points, very important. Neutrophil, they are going to eat up the pathogen. Eosinophil, they are going to be seen in allergic reaction and parasitic reaction. Mast cells release histamine. Macrophage, remember about eating again. Dendritic cell and B cell, you need to remember they are going to present the antigen to the T cells. So dendritic, directly they are going to present to the T cells, direct T cells. B cells are going to present to class 2 MHC restricted T cells. Okay, helper T cells, class 2, cytotoxic T cell, class 1. Memory cells is for future response. Natural killer cell, they don't care about the MHC. So MHC independent killing. So all of these points are highly essential for your MCQs. I want you to remember all these points. Okay. So I'm not going to discuss much about MHC here. I'm going to say only one important MCQ that you need to know. So few MCQ. MHC, what is MHC? It is going to be, it's a surface molecule, cell surface molecule, which is encoded by HLA, because yesterday we talked about HLA. Okay. Now, this MHC is of 3 types, 1, 2, and 3. So we discussed about 1 and 2. We need to discuss about 3. So before jumping into 3, you need to remember MHC 1 activates what type of T cell is CD8 T cells. MHC 2 activates CD4 T cells. 2 activates CD4, 1 activates CD8, 2 activates CD4. This again is a highly repeated MCQ. MHC 1 activates CD8 T cell, MHC 2 activates CD4 T cells. Okay, now MHC 3, this is NEAT PG MCQ. The answer for that MCQ was TNF alpha. So MHC 3 includes, so these things you need to know: soluble protein of complement system, C2 and C4, heat shock protein, TNF alpha and beta, and properdin factor B. So these 5. MHC 3 includes soluble protein of complement system, C2, C4, heat shock protein, TNF alpha and beta, properdin factor B. So these points you should know. Okay, am I clear till now, everyone? Are you all okay with the speed? We can't waste a lot of time in immune system. We have to grab the quick short points so that we don't miss anywhere in immune system. Am I clear? Yes, good. Now, going to the next. So we talked about interleukin yesterday. So I should have mentioned this first. If microbiology was started first, I would have started with this. So interleukin 1, interleukin 2, interleukin 8, interferon, tumor necrosis factor. These are the basic cytokines that you need to be aware of. So 1, we yesterday saw either acute inflammation or fever. Acute inflammation, fever. 2 means they stimulate proliferation of other T cells. Interleukin 2 stimulates proliferation of other T cells. So that's the point you need to remember. So they activate natural killer cells also. So they stimulate the proliferation, that's multiplication, division, dividing potential of the T cells. So they help us to get a lot of T cells and also activates the natural killer cells. That's what interleukin 2 do. So interleukin 8 and leukotriene B4. So these 2 are chemotactic factors which attracts the neutrophils. Yesterday we saw, I draw a picture of inflammation. So where one of the things is leukotriene B4 and interleukin 8. Okay, along with C5a, they will be useful in active acute inflammation. Next is interferons. So interferons means you need to remember they are involved in granuloma formation. Interferons means granuloma formation. That is the point I want you to remember. Interferons means granuloma formation. For example, TB. Okay, so tumor necrosis factor means it is tumor necrosing factor. So apoptosis of cancer cells. Apoptosis of cancer cells. So these are the 5 important points from the cytokines. So cytokines, what are they? Basically, they are signaling molecules, which is present during the immune response. So I hope that is fine. So cluster differentiation yesterday itself we saw, so I'm not going to talk about this. All you need to know is T cell marker, B cell marker, pan T cell, pan B cell marker. So pan T is going to be 3, pan B is going to be 19. That is the point you need to know. Okay. Now going to the next one. So complement pathways made easy. So a lot of students struggle with complement pathway. So this you need to know what is complement pathway. They are circulating proteins. So what do? Instead of, see, there is phagocytosis that is happening by our WBC. So alongside there is complement pathway can also happen. So they complement the action of phagocytes and the antibodies during the immune response. So that is what they do. So it starts with the activation of C1 and ends with C9. So C1 all the way to the C9. So in between there is something called C5 to C9, which is a complex. So this complex is called as membrane attack complex. So it will start from C1, slowly it will come to C9, C1, C2, C3, C5, C9. So once it forms the C5 to C9, they will dig a hole inside that organism. So this C5 to C9 is a membrane attack complex. If you see this, the C5 to C9 will attack the cell like a drilling machine. It will go inside the cell will swell and burst. So these are all the points that you need to know. I won't go much detail into these things. So these are just basic understanding I'm trying to give you here. So we saw something called opsonization yesterday. I explained yesterday itself. We saw something called opsonization yesterday. The C3b is called the opsonization molecule. So opsonization is nothing but tagging a pathogen. It's like you are posting a sticker over the pathogen, tagging a pathogen, and helps the phagocytes to find them. So opsonization is nothing but a sticker over a pathogen, so your immune system can easily find it. So for example, COVID-19, there is opsonization happening from C3, so it puts a sticker over that. This is COVID-19. All the immune systems, please look at here and start attacking here. So everywhere they find that sticker, they'll go and attack easily. So that is opsonization. This is just for simplification I'm saying. That's not actually how it works. Okay. Okay. So if you see, the central role is going to be C3, because most important in attacking is tagging. So if you don't tag something, you cannot attack it. So C3 plays a major role here. So this C3 can further go form a MAC, membrane attacking complex, and it can destroy the cell, that is lysis. Or this C3 can directly be involved in inflammation. This C3 is tagging, so opsonization, so that other cells can go and attack them. Okay, so this C3 is a central player, key player of this pathway, complement system. So that MCQ you must remember very well. So where does this C3 comes from? One is directly from C1, that is the classical pathway, or there is something called the alternate pathway. That is C3b, directly the C3 is coming, that is alternate pathway, or there's something called lecithin pathway. Lectin pathway. So it's from the lectin C4 to C2. So from C4 and C2 it comes. Okay, as C3. Someone's video is on, please turn it off. Okay. So you don't have to remember these points. You have to remember C3 is the key feature here. Okay. So in these 3 points, what you need to remember is classical pathway means C4 and C2. Lectin pathway is again C4 and C2. So classical lectin, no matter what, it is going to be C4 and C2. These are going to be the intermediate steps, C4 and C2. These are the intermediary complements that is happening here. Whereas in alternate pathway, directly C3. So alternate means C3. So alternate pathway directly is C3. Alternate C3. So this picture is something you must remember. This has a lot of MCQs to cover. So this single picture will help you. If I'm stressing more on these, lot of questions have come from this topic. That's why. Okay, then comes different complements and their activity. Only 3 points I like to say from this. One is anaphylatoxin, that is C3A, C3A, C5A, right? This is anaphylatoxin, C4A. So when you find A in any complement, you need to remember it is anaphylatoxin. So C4, C5, C3A, these are anaphylatoxin. Number 2 is C3B, C4B is going to be for opsonization. So when you find C3B, C4B, you need to remember opsonization. So for your exam purpose, just remember C3B for opsonization. The third point is C5 to C9 membrane attack complex, so membrane disruption. So these are the only points, 3 points I want you to remember. Okay, now we saw about engulfing the microorganism. So that is phagocytosis. So we have to know what if this phagocytosis does not happen properly. So when there is a disorder in this phagocytosis, what is going to happen? So you need to know there's something called Chediak-Higashi syndrome. Just key points you need to know. Chediak-Higashi syndrome, in this, the ability of the phagocytes to store the material in lysosomes or release their contents is gone. So which means the ability is reduced. Chediak-Higashi means ability is reduced. So recurrent pyogenic infection you will get. A recurrent infection you will get. Chediak-Higashi, reduced ability, recurrent infection. That is the first point. Chediak-Higashi, you have to remember R, reduced ability, recurrent infection. Number 2 is chronic granulomatous disease. In this, reduced production of H2O2. So in first, ability is reduced. In second, production is reduced. That is the point you need to know. Chronic granulomatous disease. So what happens here? You get fungal infection also. So bacterial, along with bacterial, you get fungal infection also. So chronic granulomatous disease. Number 3 is Job syndrome. Here, what happens? The response is reduced. Reduced response. So the response is reduced in Job syndrome. So you get recurrent abscess, recurrent cold abscess and eczema. Next is lazy leukocyte. So what happens here? Reduced movement, the migration, the movement of the neutrophils is reduced. So you just get low grade infections because it is lazy, but it will still reach. So slowly they will reach. So there's always some low grade fever the patient is going to have. Next is leukocyte adhesion deficiency, which means adhesion is reduced. We saw yesterday margination, rolling, adhesion, migration, finally. So these points you need to know. Next is myeloperoxidase deficiency, which means reduced acid production, HOCl production. So the killing is going to be delayed. There is going to be killing, but the killing is going to be delayed. So all points are important. They are all MCQs. Am I clear till now? Is everyone clear? Very good. Next. I am skipping this point, acute versus chronic inflammation. We talked a lot about acute versus chronic inflammation, so I'm skipping this point. Then, graft and their rejection. So you need to know there are 4 grafts: autograft, isograft, allograft, xenograft. So one part of the body to another part of the body within the same individual is going to be autograft. So one human to another human is going to be either isograft or allograft. But xeno is between 2 species. So do not expect a question from xenograft. You are going to get questions from these 3: auto, iso, and allo. So iso means twins. Iso means iso twins. Allo means between 2 different members. Allo means between 2 different members. Iso means twins. These are the only 2 points you need to know from here. So what are we going to do? Organ transplantation. If we transplant one organ to another, from one organ to another organ from the same person, that is autograft. From twins, that is isograft. From 2 different people, that is allograft. Okay. Then we saw about the reactions, transplant rejection. So transplant rejection. So you have to know it can be acute, it can be hyperacute, it can be chronic or graft versus host, which I told yesterday itself. So hyperacute is the one that occurs immediately. Hyperacute occurs immediately. Acute takes time. Chronic takes more time. Graft versus host, you can't actually say. In graft versus host, you cannot say the time. So what you need to know from here is hyperacute is type 2 hypersensitivity reaction. Acute is type 4 hypersensitivity reaction. Chronic is type 3 and type 4 hypersensitivity reaction. Graft versus is type 4 hypersensitivity reaction. So chronic you can follow type 3. So chronic follow type 3. Type 3 hypersensitivity reaction for chronic. Graft versus host you can follow type 4. So acute and graft versus host you can follow type 4. Hyperacute type 2. Chronic is type 3. Am I clear in this topic? Very good. Okay. Now going forward. So immunoglobulin I am not going to talk because this is what we talked yesterday. Hypersensitivity we talked yesterday. So regarding the other names, this is what we missed yesterday. I think we didn't have time to cover that. So the names you need to know, type 1 is anaphylactic type, that is immediate type. Type 2 is cytotoxic type. Type 3 is immune complex type. Type 4 is delayed type. Okay. Type 5 is stimulatory hypersensitivity. Stimulatory hypersensitivity. So you need to know type 1, 2, 3 and 5. So you can even skip 5. Type 1, 2 and 3 are antibody mediated, whereas type 4 is cell mediated. This point is very important. Type 4 is cell mediated. Okay. This point is very essential. So note these points, these are essential. Type 1, anaphylactic or immediate. So type 1 is immediate, type 2 is cytotoxic, killing the cells. Type 3 is immune complex, type 4 is delayed type. So we saw a lot of examples. So you will have to read much more examples. Next is immunodeficiencies list. List of immunodeficiencies. So you need to know when the T cell is gone or the B cell is gone, when the T cell or the B cell, when they are gone, what is going to happen? So when the T cell is gone, you are going to get something called Dye-George effect. Dye-George effect. Okay. So for this, I would like to give a mnemonic, that is, so if you miss this in exam, you will feel a lot. So you have to memorize right away. When the lecture is happening, you must memorize. So I will provide mnemonic here and there. So George die in Taiwan. George dies in Taiwan. So this is what you're going to remember. So George die, which is Dye-George, means Taiwan. That is T cell. So this is essential. Next is B cell. B cell defect are going to be these three. In this table, you don't have to remember the defect. All you need to know is the names that are coming, syndromes that are coming under the defects. So B cell defect means X-linked A gamma globulinemia, common variable immunodeficiency, and immunodeficiency with hyper IgM. So this you need to memorize. B cell defect means X-linked A gamma globulinemia, common variable immunodeficiency with hyper IgM. Okay. So I think I can give a mnemonic for this also. I just thought of one, maybe it may be helpful. So this is B cell defect. So in local terms, you can use a word, that is sorry, bunyan. Remember the bunyan that we use, the bunions. So there are two things that are very commonly selling or most selling in bunyan, two sizes. One is XL size, another is medium size. Just remember this point. XL size, medium size. These two are very common. So these two are very common. So common stands for common variable immunodeficiency. XL stands for X-linked agammaglobulinemia. Medium M stands for IgM, hyper IgM. So IgM, XL, these two sizes are very common. So in bunyan, these two sizes are very common. Can you remember this? Is this fine? Okay. Next is combined B cell and T cell defect. The other syndromes: Nesloff syndrome, ataxia telangiectasia, Viscott-Aldrich syndrome, severe combined immunodeficiency. So if you rule out this bunyan and this George, anything else is going to be combined immunodeficiency. So XL and M are common in bunyan. So B cell, common, XL and M, IgM. Okay. So X-linked agammaglobulinemia and IgM, and common variable immunodeficiency. George dies in Taiwan. So Taiwan, why I said, because it is thymic hypoplasia, which is a very important MCQ. In Taiwan, a lot of people will be short. So thymic hypoplasia, you need to remember what happens, hyperplasia or hypoplasia. You have to remember it is hypoplasia. In Taiwan, every people is short. So the thymus is short. So hypoplasia in thymus, that is Dyegeorge syndrome. That is a highly repeated MCQ. And Dyegeorge syndrome is a T cell defect. That is second highly repeated MCQ. So in this entire table, if you take one point which is essential most, means Dyegeorge syndrome. So you should never forget this point. Okay. I'm stopping again here. I want to reconfirm. Am I clear till now, everyone? So will you be able to make all the questions from immune system? Because if you don't get the concepts in immune system, you cannot sort it out. Okay, very good. Now, I can skip this infection that can cause immunosuppression. That's not important. Miscellaneous test. So test and all we will see anyways. Sterilization, disinfection, we will see anyways.